Hey there folks, and welcome to the first video for Chapter 10, Molecular Biology of the Gene. In this chapter we're going to be taking a closer look at DNA, how exactly it is built, and how its code functions to store our genetic material, as well as some of the history of how we discovered DNA in the first place. Speaking of which, molecular biology is essentially the field of studying genetic heredity. This is not the exact same thing as genetics because molecular biology focuses on the physical construct of DNA itself, whereas genetics tends to focus on the functioning of our genes, how frequently we see them, and what sort of traits they code for. In molecular biology, we focus on the physical DNA, what it's made of, what those parts are made of, and how DNA functions as a molecule. Early on, early cell biologists and sort of proto-molecular biologists thought that our genetic material was coded in some kind of protein system. However, in the 1950s, we discovered the existence of DNA as the record-keeping language for our genetic information. And in 1952, a very important series of experiments by Martha Chase and Alfred Hershey cemented the existence of DNA and its function in our cells. When we talk about DNA, and RNA as well, it's going to be very useful to first have a solid understanding of not just what it looks like, because we'll get into that in a moment, but what it's even made of to begin with. Imagine DNA as a language which can express very long, complicated ideas, but which doesn't actually have a lot of letters to use. Specifically, DNA only has 4 letters it can use: C, T, G, and A. This is because DNA and RNA are what we call polymers of nucleotides. These nucleotides are essentially the letters that make up our genetic code. One nucleotide consists of a 5-sided sugar, that's here on the left, a pentose sugar, which has a phosphate group on one side and a nitrogenous base on the other. And what separates the different types of nucleotides is exactly which base is attached to that sugar. DNA and RNA each have 4 nucleotides. There is one nucleotide that appears in DNA and not in RNA because RNA replaces it with something else. In DNA, the 4 different nucleotides we have are cytosine, guanine, thymine, and adenine, and they only form bonds in specific pairs. Cytosine and guanine will only bind to each other. Thymine and adenine will only bind to each other, and never the other. In RNA, a nucleotide called uracil replaces thymine, and it binds to adenine. Adenine will never bind to cytosine or guanine, and so on for all of the other bases. They only ever appear in their specific pairs. And so we can even measure DNA and RNA in terms of nucleotide pairs, because it's very specific. That is our usual measurement for how long a DNA or an RNA molecule is. When I say that DNA and RNA are polymers, by the way, this is a term from chemistry. A polymer is a large molecule that is made up of smaller connected molecules. In this case, DNA is a polymer, a very, very large one, and the smaller molecules that bind together to make it a polymer are these nucleotides, these nuclear bases. The sugar, that pentose sugar in DNA, is called deoxyribose. Any molecule you'll hear of that ends in ose is a sugar, whereas in RNA it's a smaller, simpler sugar, still has the same 5-sided shape though. In RNA the sugar is called just ribose instead of deoxyribose. The shape of DNA is something that was an area of study that was a mystery to molecular biologists for quite a while. However, after Hershey and Chase discovered DNA and cemented the fact that yes, it is our way of storing genetic information, it became one of the most important areas of study in all of biology. And by 1953, we had finally discovered the shape. By the way, I say finally, but that's 1 year between discovering and confirming DNA and discovering its shape. That's pretty awesome, if you ask me. Discovering the shape of DNA is attributed to 3 different scientists, Rosalind Franklin, and a research team constructed of James Watson and Francis Crick. But there's something you should know about this team, which is frequently not covered enough in many textbooks. The formal discovery of the shape of DNA is frequently attributed to Watson and Crick. But that's not entirely correct. Watson and Crick were studying the shape of DNA, and their ideas were, by majority, incredibly incorrect. They contacted another researcher they knew, Rosalind, who had been taking photos of DNA. She had become a master of a particular type of molecular photography called X-ray crystallography, which allowed her to photograph the shapes and structures of molecules. At the time, this was much more difficult to do than it is now with our modern day technology. I say modern day, but this is only about 70, 80 years ago. At the time, though, this was the best tool that they had. They asked her to look at DNA, and because of the work she had already been doing with X-ray crystallography, when she took photos of DNA, she was immediately able to recognize exactly what the structure of DNA was, and she was the very first scientist to ever do so. The problem with discussing Watson and Crick is this. They were eventually awarded the Nobel Prize for Science for their discovery and publishing their discovery of the shape of DNA. Rosalind Franklin's role in the discovery, however, went unacknowledged for many years, and in fact, Watson and Crick stole the photos and the information that she made. What I would like you to know about this discovery is that eventually Rosalind Franklin was acknowledged, but it took far too long to do, far, far longer than it should ever have taken, in part because she was a woman, and at that time women in biology, as well as in any other form of science or medicine, were quite unusual, and she was disrespected because of this. You will know better. It remains an area of molecular biology's history that is not covered the way it should be. So I would like you to know very clearly that Watson and Crick had been studying DNA, and they were going after what shape DNA has. And they were wrong. If you ever find examples of their old models of what they thought DNA looked like, they were way off. It's kind of amazing. And remember to pay proper respect and give credit to the work that Rosalind Franklin did. As you can see here on the right, this is the shape of DNA as we now know it. DNA has a double helix shape, which means that the shape of DNA is 2 spirals wrapping around each other, stretched out, as though you were taking a ladder and you just kept twisting the ladder until the sides of it went round and round each other, but you still had the ladder rungs connecting the 2 sides going down the center. In DNA, these rungs are formed by the bonds between those nuclear bases we were just talking about. RNA is 1 helix. It doesn't have 2 sides, it's just 1 strand. And going down around the center of the inside of that spiral are the 4 bases that make up RNA. Cytosine, guanine, and adenine are the same between DNA and RNA. But remember, uracil replaces thymine in RNA. When our cells make new DNA, the new copies of DNA are what we call semi-conservative. And if you'll forgive the joke, this has got nothing to do with their political views. What it means is that in each new copy of DNA, one strand comes from the original copy and one strand is new. In every single copy, when our DNA is being replicated, there are molecules that attach, enzymes that attach into the old strand and break the bonds, break the ladder rungs that are holding the two sides together, so that the DNA can open up and essentially forms replication bubbles. In these bubbles, replication goes on, building a new strand of DNA to attach to that open area, until finally all the bubbles open up and connect with each other, and the two new copies of DNA float away from each other. Here you can see DNA polymerase, which is an enzyme that holds the one side, the old side of the DNA, as a scaffold and pulls in free nucleotides and attaches them where they fit to the old strand. So say if DNA polymerase is reading the old strand and it reads that here is an adenine, it will pull in a thymine to attach to it and complete that bond. And it goes all the way down the length of that bubble until that bubble hooks up to the one next to it. Helicase is an enzyme that you see right there at the center of the replication fork that breaks the bonds like a knife, cuts the bonds, and opens up the replication bubble so that DNA polymerase can follow along behind it on either side, building the new side of that semi-conservative copy of DNA. When we look at DNA replication, by the way, we have specific terms that refer to which strand in the new DNA copy you're referring to. The leading strand is the brand new strand that DNA polymerase is building. The lagging strand is the original side that DNA polymerase is following as a model. Alright, folks. This is going to wrap it up for the first video of Chapter 10. The next one is going to be a long one, so take a little breather maybe. Alright, we're going to get into how we get from storing our DNA in that 4-letter code to actually breaking it out into the cell, and the first steps toward really using our DNA. But I'll see you doing that in the next video. See you next time, folks. Bye.